የዛሬ እንግዳችን በማወያየትና በመምራት ዶክተር ፋሲል ተኮላ-አየለ ይባላል። ዶክተር ፋሲል early stand investigator ወጣትና early stand investigator በናሽናል ኢንስቲትዩት ኦፍ ሄልዝ ተመራማሪ ነው። ይሄ ፖዚሽን በጣም ፕሬስቲጂየስ የሆነ ናሽናል ኢንስቲትዩት ሄልዝ ለወጣት ተመራማሪዎች የሚሰጠው ነው። ፋሲል በመጀመሪያ ዲግሪውን በደቡብ ዩኒቨርሲቲ ከዛም ኤምፒኤች በአዲስ አበባ ዩኒቨርሲቲ ከዛም በመቀጠል በፒኤችዲውን በጄኔቲክ ኤፒዲሞሎጂ በዩኒቨርሲቲ ኦፍ Brighton and Sussex in the UK, United Kingdom. ሰርቷል። ከዛ በመቀጠል ፖስትዶኩን በዚህ በዩናይትድ ስቴትስ ናሽናል ኢንስቲትዩት ኦፍ ሄልዝ እና በተለይም NHGRI ናሽናል ሂውማን ጂኖም ሪሰርች ኢንስቲትዩት ሰርቷል። ፋሲል ሰፋ ያለ ልምድ ያለው ነው በጄኔቲክ ኢፒዲሞሎጂ፣ ግሎባል ሄልዝ፣ ትሮፒካል ዲዚዝ እንዳውም በቅርብ ጊዜ እንዳውም በዋናው በአሁኑ ጊዜ ደግሞ በቻይልድ ዴቨሎፕመንት እና ካርዲዮ ታቦሊክ ዲዚዝ ላይ የሚሰራ ተመራማሪ ነው። ፋሲል ከ100 በላይ ፓብሊሽ አድርጓል። ከ100 ሳይንቲፊክ ፒር ሪቪውድ in a high impact journal. One of his really impressive work was published in New England Journal of Medicine. I don't know how many of you heard about this journal. This is the tip of the iceberg. Like the impact factor is 91.24. When I checked last night, it was 91.24. So you can imagine how impactful his contribution to the field is so far. So he's at his very high trajectory. So today he's going to talk, as you can see in this title, he's going to talk African genome research, examples and potential to improve public health. So this is a very good coincidence actually, with timing is very perfect. This is a perfect time for public health. As most of you know, Bahir Dar University is launching Master of Public Health in MPH in Public Health Genomics. So, which is really a congratulative one in the School of Public Health at Bahir Dar. So this is a way to go to the field of precision medicine, and as you will learn more in the coming in the Fasil's talk, public health genomics is really a very multidisciplinary field. You can from public health to genomics to all kinds of areas of it's mostly considered one health approach. So, as I heard, Bahir Dar is accepting or trying to accept admission for students in public health genomics. So, most of you, I hope you will join this program, and hopefully we'll have a picture that looks like Ethiopia in the next meeting. So, I'll stop here sharing and let our young investigator Fasil to take the stage. Thank you, Fasil, again, and everybody, welcome to Fasil.
Okay. Can you see the screen okay now?
Yeah, maybe again swap it to. Yeah.
I have to swap?
Yeah, not yet.
Okay. Is it fine now?
Not yet. You haven't. Yeah, click there, and maybe swap. You haven't done yet.
about now?
No, it's like the previous one. Maybe unshare and try to share it again.
Okay. Okay, it's unshared already, right?
Yeah, yeah.
Sure. Okay. Okay, let me do it. Just give me 1 second.
Yeah.
Okay, how about now?
Yes, now it's perfect. Thank you.
All right, thank you. Okay, I would like to say thank you to Professor Tesfaye for the kind introduction. And I would also say thank you to Bahir Dar University for inviting me to speak and for facilitating this seminar series with Professor Tesfaye. Okay, I'll just spend a couple of seconds mentioning about my current research program. So currently I work at the NICHD, which is one of the 27 institutes within the National Institutes of Health. And I lead a research program there. And my research group focuses on genetic epidemiology of early growth in the context of understanding links between early life health and later life cardiometabolic diseases. I have had the privilege of working with and mentoring some postdocs, as well as graduate students and summer interns that have successfully moved to academia as well as government after completing their fellowship. So coming quickly to today's talk, my talk today is focused on genomics in Africa. And my goal here is to highlight the relevance of genomics to public health in Africa, and to kind of highlight the huge yet untapped potential, especially on the continent and as well as in Ethiopia. And I'll try to provide some examples from Africa and some of the work from Ethiopia that we as well as others did to try to understand the genetic population structure of the population as well as its relevance to some diseases. So I'll start with kind of slowly describing what I mean by the genome and how the study of the genome, genomics, can help in facilitating public health. So let me start with the obvious. Humans are complex on many levels, and we are all the same species, but we are all different. Our environmental interactions as well as our genome sequence are distinct. And we each have more than 30 trillion individual cells and over 200 different types of cells. And the instruction to our development and biological function is coded and governed by our DNA, the deoxyribonucleic acid. So our DNA contains 3 billion base pairs of these nucleotides, and the collective sum of all these nucleotides is what we call our genome, the genome. This is effectively the governing instruction set, and it's a regulatory code that kind of instructs us how proteins are coded, how genes are expressed, and in general, how our function and development is maintained. So what does the genome look like? So DNA is a linear molecule. It's arranged in a double helix. It doesn't look like this, but it's comprised of 4 base pair nucleotides: the adenine, cytosine, guanine, and thymine. And the sequence of these letters, A, C, G, T, that we call the genome. And we have 3 billion pairs of these throughout our genome. So this sequence is important if it determines, as I said, what proteins are produced and the amount produced and whether the protein is functional or dysfunctional. So that's why we say we are the same at the same time. Because of the sequence of proteins, we are also different. So I would like to emphasize here that the more related two people are, then the less different their sequence is. For example, a mother and her daughter are about 50% alike in the sequence. But at the same time, I would also like to mention that there is more genetic variation within human populations than between human populations. However, we define population based on ethnolinguistic levels, geographic affiliation, and others. If we have 2 random individuals from any one group, they are almost as different or as similar as any 2 random individuals from the entire world. So this is an important area to keep in mind. So why are you interested in the genome? That's because many diseases involve multiple genes. And these multiple genes interact with environmental factors, resulting in functional issues that lead to diseases. So there are some diseases caused by monogenic, they are called monogenic diseases, where a few genetic aberrations are consequential, which means they result in those diseases. But for the field of public health, where we are interested in common diseases that are prevalent across the population, genomics interacts with the environment to result in the disease. So it's not like the genetic aberration will immediately lead to a disease, or the environmental factor or exposure alone leads to a disease. It's the interaction between the genes in the environment that lead to a disease. So we should keep that in mind as well. So because of these, we have a dedicated discipline known as genomics that studies the genome and health. It's relatively new, but it's also a rapidly expanding field of science. It's quite multidisciplinary. Fields from epidemiology, statistics, biochemistry, genetics, molecular biology, bioinformatics, and others come together to describe and understand biological information encoded in the genome. So many examples of the public health benefits of genomics have already emerged in developed countries, and that's rapidly accelerating as well, whereas we are yet to see its application in low-income countries. I'll talk about it later. So I would like also to say a little bit about what the history of the field. So we can go earlier than what I have here, but the 1975 to early 90s was foundational to genomics, because scientists established some protocols for isolating genes known as DNA cloning. They also developed protocols for amplification, which means we can copy portions of the genome so we can study them use them in different studies, sequencing and bioinformatics as well. As I say, these are very, very foundational, but these were the era when only small segments of the genome could be studied at a time, because it's very difficult to study the entire sequence of 3 billion pairs. But the year 2003 marks a historic scientific achievement. The Human Genome Project was completed after 20 years of vigorous work by an international team of scientists. And what the Human Genome Project did was it provided the first near complete sequence of the human genome. That's the blueprint of the human biological information, as some call it. And I joined the genomics field after the Human Genome Project was completed, and more senior people who have been in the field for many more years before I joined the field tell me that, oh, you're very lucky, because here we have the reference sequence that you could use and interrogate to ask different questions. And the year 2005 is also foundational, because that's when next generation sequencing technology emerged. And with that, we mean it allows generations of many millions of DNA sequence short reads with faster output and with decreased cost. That's also fundamental to make genomic sequencing services accessible by many laboratories. So in the past 20 years, since the release of the first complete sequence of the human genome, the use of genomics has increased in medicine, public health, and other fields. And it's a good memory to all of us. Recently, with the COVID-19 pandemic, it has focused intense global attention on the use of genomics, both to diagnose infection, to do surveillance of the infection, as well as in identifying mutations of the SARS-CoV-2, as well as making public health decisions. So what are the applications of genomics in public health? One is public health screening. So it's useful to advance global burden of disease surveillance, as well as food security. As I said, in the case of COVID, pathogen surveillance, and it also helps to provide tools for medical care. Carrier screening, it's practiced even in some African countries, particularly neonatal screening, although the program is limited in many sub-Saharan African countries, but some countries, Northern African countries, also do newborn screening to diagnose some developmental disorders. Pharmacogenomics is another application of genetics, where we use data from the genome, from DNA, to inform drug development and testing, but also in terms of precision public health to correlate individual genetic variation with drug response, and to correlate appropriate drugs to different individuals based on their genotypes, as well as other factors. Health disparities is socioeconomic difference in distribution of disease and health outcomes or access to care, and population genomics can play a role in understanding the genetic causes of disease in different populations, especially in becoming part of the solution to health disparities by making genetic services and genetic discoveries across all populations. Okay, the biomedical impact and application of genomics is numerous, and arguably, I think several scholars say that genomics advances have provided more insight than any other life science research in the modern age. This is a figure from Tekola-Ayele and partners summarizing some of the functional impacts of genomics on human health and clinical practice. So I will not go through it, but the numbers 1 to 8 are a summary of the different areas of applications, including public health, in medicine, technology development, therapeutic advances, every field, environmental genomics and metagenomics, gene editing, so human microbiome interactions, showing that it has huge impact on our everyday life. So I would like to next to hint on the relevance of including Africans in large-scale genomic studies from the perspective of disease burden, etiological studies, as well as in clinical decision-making. Why genomics in Africa? Why do we need genomics in Africa? One, which is very obvious, is that there is a huge disease burden in sub-Saharan Africa. There are population differences in the burden of chronic diseases. We know that infectious diseases are prevalent, non-communicable diseases are on the rise, and most of these diseases result from the interaction of genetic and environmental exposures. And we know that most recent centuries in Africa have been characterized by extreme changes in the environment and lifestyle, including changes in diet, behavior, urbanization. And this has implications. One, it triggers risk to disease on a background genetic susceptibility. As I said earlier, most common chronic diseases result from the interaction of genetic and environmental factors. So the change in the environment helps trigger the background genetic risk. So with the rapid change in the environmental factors and lifestyle in Africa, it makes it very relevant to have genomics. And secondly, I'll also mention it later, the environment, not recent change in environment, but the environmental changes over the years, maybe tens of thousands of years, when the continent environment was transformed, could impact change in the variation distribution. And this would result in selection of some variants that are relevant for survival, but some of these could be disadvantageous when the environment changes or when people move to a different setting. I'll give an example later, such as when people of West African ancestry move to the Western nations and are exposed to these factors that predispose to chronic kidney disease. Second reason why we need genomics in Africa is Africa holds the largest genetic diversity. This is unrivaled by any other continent. Here, genetic diversity or genetic variation means rich coded information. And that information can be beneficial on many fronts. As the topic of my talk, it is very beneficial to understand disease etiology and to inform public health. But genomic technology also increases economic development. So with Africa holding the largest genetic diversity, this means there is a lot of unmined potential and information for innovation development as well as for public health. And third reason is discoverability of new genetic variants is higher in Africa than in other populations. There are many reasons to this. This is because the number of variants that differ from the reference sequence that I mentioned was first drafted when the Human Genome Project was completed is much higher in African populations. For example, with the recent AfRIE Africa work, just with 420 something genome sequences, they were able to identify more than 3 million new variants that were not found, that didn't exist in the reference. I'll also talk about our work in the African Genome Variation Project, where we discovered more than 8 million new variants by including just a sequence of 100 people from Africa, especially from Ethiopia and South Africa. So discoverability of new variants is higher. This could have different reasons because of population history and demographic events. Both archaeologists as well as geneticists agree that humans lived the longest in Africa before migrating out to populate the rest of the world. That led to a chance for increased diversity and mixture, environmental challenges. So new mutations arise, and all the mixing also results in new patterns of variant distribution. So without studying people in Africa, we cannot identify those variants because people from other continents carry only a subset of those genetic diversity of the continent due to a bottleneck event that happened during the out of Africa migration across the northeast, the Horn of Africa. So an important breakthrough, I would say, in terms of characterizing the genetic variation landscape of African population is this work by Sarah Tishkov, published in Science in 2009. They studied more than 120 African populations, along with non-Africans, using a type of genetic markers known as microsatellites. And they found that there are 14 ancestral population clusters in Africa, and that correlate with shared culture and linguistic properties. They also found, this is very important, high level of mixed ancestry in most populations. So most populations are mixed. You can see the figure at the bottom, with the stacked bar plot, and each vertical line is one individual, and the different colors represent one of the 14 ancestries they detected. So you see, no individual has a single color along the vertical axis, which means the majority of people have mixed ancestries across the continent. So after the Human Genome Project was completed, high throughput technologies enabled genome-wide assaying of SNPs, single nucleotide polymorphisms, beyond microsatellites used in earlier studies. So a couple of international efforts have established to characterize this human genetic variation. One of this is the Human Genome Diversity Project, but two of these are the HAPMAP and the 1,000 Genomes Project. The HAPMAP identified 10 million variants in 11 populations, and later the 1,000 Genomes Project made a deep sequencing of 26 global populations and identified more than 80 million variants. So these are great efforts. They involved people from different parts of the world, but still, compared to the ethno-linguistic, geographic, and environmental diversity of the African population in the continent, the populations represented in this project were by far fewer than the genomic variation landscape expected for the continent. So about 9 years ago, we formed the African Genome Variation Project through a consortium of research institutes in Africa and the US and the UK. The goal was to perform dense genotyping of diverse ethno-linguistic groups to give new insight into the genetic variation landscape of Africans and to facilitate genetic epidemiology studies. So 20 ethno-linguistic groups were included, and from each group, about 100 samples were either sequenced or genotyped using arrays that help us type markers of up to 2.5 million variants. It was published in Nature in 2016, and you can see there are population groups from Ethiopia as well. Not the good representation of the entire Ethiopian linguistic diversity, but some population groups were included there as well. So what are the findings? One, genetic diversity is highest among African populations than European or Asian populations. This evidence has come from the number of variant sites per person and by measuring the genetic differentiation between pairs of individuals. And second, there are by far more genetic ancestries within African populations than other populations. So if you combine other the total number of ancestries in other continents together, the number is around 12. But per continent, within African continent, we were able to kind of see about 11 ancestries. So we also identified new genetic variants, about 8 million, that were not there in previous global efforts. And the plot that you see is an effort to produce a more refined African population substructure. And it's called the top 2 genetic principal components, where you decompose the thousands and millions of markers into few polars and see how people cluster with each other and away from each other. So on the first principal component on the x-axis, you see that East Africans, which is basically Ethiopian ethno-linguistic groups I mentioned earlier, they cluster separately from West and South Africans. That's the first observation. And then the second principal component, if you see along the vertical axis, you can see that West Africans separate from South African populations. I don't want to go in detail into this, but here what we see that the correlation between genetic markers that are close together is weaker among African populations than among Asian or European populations. So we call it quick, rapid decay of correlation, or call it haplotype or LD. So as you can see in the figure, Africa is at the bottom, which means correlation is weak and correlation decays faster among Africans. What is the relevance of this? This is very important because it facilitates discovery of causal variants. I would like to spend 1 minute here. It's an advantage, a fourth advantage of why African population genetics is important. Genomes of Africans are useful to pinpoint disease-causing genetic variants in many instances better than genomes of European or Asian populations. Why? Because, as I said earlier, the group of DNA variants that are inherited as a block from a single parent are in general shorter in African genomes. And I tried to put a cartoon here where you see in the European ancestry a very long block, whereas in the African ancestry it's broken down into pieces. This is because African populations lived longer, which means there were a lot of events for recombination of these sites, which break down the haplotypes. So we have shorter segments. And there was a lot of population movement and mixing that also contributed to this. This is advantageous from a scientific and discovery point of view because it helps to pinpoint the causal variant. In the first case, if any association is found between a disease and any of those blue markers, or then we wouldn't know which of the blue or markers is causal or the red marker is causal because the block is long. Maybe it has tens of thousands of variants, so it could be one of those, but we couldn't know. But in the case of African populations, usually it's shorter, so we can narrow it down, and that's very helpful to do functional experiments and move towards translation of the findings into clinical and public health impact. This has global health benefits. Even if a variant is discovered in other populations, we can bring it down to African population genomic studies and pinpoint the variant, and that is helpful to every human population. So this has been done with TCF7L2 and type 2 diabetes. And recently we did the same approach for variant linked with fetal growth by using African Americans in the US. So why the Ethiopian genome? Again, just like my argument for including African populations, disease burden is one of the reasons. But in addition to that, Ethiopia is also at the epicenter of human migration, both out of Africa, the out of Africa migration of human populations who lived in Africa for years and before they populate the rest of the world. The route of migration, it's argued, but it's through the Horn of Africa, through the north or south route. That's important. But also back to Africa migration. Human beings migrated out, and there was also historic archaeological evidence for back to Africa migration, both because of trade and other relationships, and because of a lot of contacts that the Horn of Africa had with the rest of the world. So what happens during this? Out of Africa, bottleneck events, mutations are created, not all of them are moving out. So, and back to Africa, admixture on a global scale. So these are all have implications on health. So, so studying the Ethiopian genome can be helpful to inform all these events and understand its impact on disease resistance as well as susceptibility. The ecological dimension is the other reason. So we have the extremes of the ecological and geographic dimension, which shapes the genome, and that's important. Cultural and linguistic diversity is also essential to understand the African variability by studying Ethiopians. But the earlier studies focused only on mitochondrial and Y chromosome, which is limited. So a relatively comprehensive study of the Ethiopian population genetic variation was done by Luca Pagani and his colleagues, including colleagues from Addis Ababa University. Here, 235 people from 10 Ethiopian and 2 neighboring regions were genotyped. So I would like to take a step back and say, bear in mind that these are not, again, representative samples of the country. And I would also like to add that where the samples were obtained might have bias on the report. I'm not saying the report is biased, but the interpretation of the report. So take it as a pinch of salt. 2 key findings from their work. 1, there is genetic variation within and between population groups. So remember the within as much as the between. So sometimes the between is emphasized, but data shows that self-identified sub-ethnic and ethnic designations do not reflect genetic ancestry. There is a lot of mixed ancestry among people who self-identify as belonging to one group. This is true for the US. This is true globally. I'll mention a study we did later where we found that more than 97% of human beings across the world have at least 2 ancestries, genetic ancestries, not just one. So it's also strongly true, especially for Ethiopia, because there has been a lot of admixture events, not just within the people within the territory, but also globally because of its historic interactions with people around the world. So, so it's important to also note the sampling and often the disconnect between self-identified ethnic group and genetic ancestry is neglected. So it's important to keep that in mind. The African and non-African haplotype components, they also found that there are African haplotype components and other components they called non-African, but refined work is needed what that really means. Is non-African, European, what is it? So I'll talk about it as well. So in another study we looked at samples from the Walaita zone in the context of broad continental ancestry from the HAPMAP. The HAPMAP is a project that has 11 other populations, including some from Africa, like the Maasai from Kenya, Luya from Kenya, Yoruba from Nigeria, and African Americans from the US, as well as other Europeans and East Asian, as well as mixed Hispanic populations. So So just pay attention to the left extreme plot, which belongs to the Walaita from Ethiopia. So you can see three colors. So each vertical line represents 1%. So the three colors represent three major ancestral components. Pink, the top part, is the major proportion which we do not see much in non-Ethiopians. In another study that I'll talk about later, this appears to be the omotic ancestral component. And purple is the West African ancestry. With further refinement, this appeared in another study, because this is a broad continental study. And green is another East African ancestry that is also seen in the Maasai and Luya from Kenya. Perhaps it's lowland Eastern Cushitic ancestry. And you see tiny yellows here. It's yellow is major European ancestry. And you see very insignificant or negligible amounts of this in the Walaita, and also in most other Ethiopian population groups. So I'll come back to the relevance for clinical decision later in my talk. So we continued working on refining this, as well as trying to see where how many global ancestral components there are in global populations, including in Africa, led by my former colleague Daniel Schreiner in Charles Routin's lab. So we assembled the largest global genomic data sets, and we found 19 ancestral components globally, and 7 of these are predominant in Africa. So with regards to Ethiopia, bear in mind again that only a few samples and linguistic groups were studied. We haven't done new data collection. We used existing data. So there are two key findings. One, the Ethiopian samples have 4 or 5 ancestries. So that means we shouldn't model them by a two-way admixture. It's very complex kind of ancestries within the Ethiopian samples. That is the East Cushitic, Nilo-Saharan, Ogoric, Arabian, as well as a small amount of Levantine, Caucasian ancestry. Second, the omotic component, as I mentioned earlier, is limited to Ethiopia. It's mainly in the AfRIE as well as in the Walaita. And these results indicate that that's a second important point. And we didn't see the West African ancestry in the Ethiopian samples. So that led to the second point, where we said the commonly used West African and Euro-European samples. If you think about the HalfMap project and most other studies, including 1,000 genomes and others, we use the West African sample from Nigeria called the Yoruba as a reference for Africa, and the CEU from northeastern populations in Utah as a representative of the European population. So, but these two populations are not optimal choices as parental populations of Ethiopians. So that shows that further work is required to understand and use the Ethiopian genome diversity for disease as well as public health relevance. Okay, enough with the population genetic diversity topic. I want to shift this a little bit to my next goal, to bring your attention to why we do genomics for health. Migration, gene flow, selection is an important factor here. I want to highlight selection in the context of its relevance to public health. Environmental pressures in the past could have selective advantage. Let's say tens of thousands of years, only a few people with a genetic variant that favors survival against the environmental adversity survive. They reproduce, then over the long term, that specific variant will be more prevalent in the population. So that's advantages in the past, that environment. But when the environment changes, like lifestyle, diet, behavior, or people move to other places, those selective environmentally pressured selected variants could be harmful. For example, they could increase risk for another chronic disease. So the impact of the environment in shaping the genome is huge in Africa. This has been shown by several studies because Africa has a latitude spanning 70 degrees that covers a very heterogeneous environment. It's not a west-east axis as in Europe or Asia. So there is enormous difference in the climate and environment that shapes the genome as well. So infectious agents, highland adaptation, several studies in the Ethiopian highlands finding selective signals and complementing findings in other parts of the world, in the Himalayas, for example, showing what they call convergent evolution, where those variants originated and so became selected differently. So that's one. Pathogen diversity, diets, all these shaped the genome in some instances very relevant to disease. For example, highland adaptation helps us to understand hypoxia, chronic disease in birth weight, which is an area of my interest, why do and preterm birth. So these are important areas where the Ethiopian population could be useful to understand physiological as well as pathological issues. So just to give a couple of examples of discordance between the old genes, old environment, old genes in new environment, which means old genes created because of pressure by the old environment that become disadvantageous in the new environment. So this paradox has been shown in the African diaspora. So an example is the ApoL1 G1 missense haplotype. So the variant was found to be associated with up to 7 times increased risk for kidney failure in African Americans with high West African ancestry. And that leads to kidney transplantation failure and huge kidney disease disparities between African Americans and whites in the US. When we look at the distribution of this variant, it has the highest frequency in West Africa and Southern African populations, and populations of the African diaspora in the West, in the US and Latin America. Its absence in Europe, Asia, we have looked at the variant distribution in Ethiopian population. It's not present in Ethiopia as well. So why are the variants more common in West Africa? It's considered that several studies show that it has become more common in West Africa because in the past it confers protection against the deadly form of trypanosomiasis, the African sleeping sickness, which is more prevalent in Africa. So that variant was selected because it provides protection against trypanosomiasis. So people with that variant survive. So the variant's frequency becomes higher in West Africa. So when people move to other environments, or even in West Africa when lifestyle changes, but that variant becomes a problem for the renal function. So advantages in the past becomes disadvantages in the present. So I don't want to spend more time on this, but I want to say that the global disease risk variant distributions also vary. So these are just a plot of the more than 2.5 million variants in genotyping area host populations. European, African, you don't see any differences. We see a U-shaped curve, which is expected, which means low frequency variants are higher in every population. But what happens when we superimpose a selected list of genetic variants associated with cardiovascular disease, which is an area of my interest, including type 2 diabetes, dyslipidemia, and obesity. So we see bell-shaped curve for these variant distributions. It's neat in the Europeans. But if you look at the Africans and East Asians, it's less likely to be bell-shaped. Why? Because there are potential differences in the genetic architecture of disease. And another reason is there is lack of discoveries on common genetic risk variants in non-African and non-European populations. So we cannot use data from the European populations to infer disease etiology in African and East Asian populations. So there are several examples like this. Another example is hypertension, selection-driven genetic risk to chronic diseases. There is some hypothesis in the past that genetic susceptibility to hypertension is ancestral and it's due to latitude heat adaptation, which means individuals who are very close to the tropics are likely to have that variant, which leads to increased risk for hypertension. So supporting this, we found in the African Genome Variation Project that latitude could be a selective factor for a hypertension risk variant. The tyrosine protein kinase gene increases, the variant increases as we move towards the equator. So differential susceptibility to hypertension potentially is due to adaptation to temperature and latitude. I would like to add an example from Ethiopia. In a study that we used samples from the Walaita zone, we discovered signals of selection on a gene called the PPARA, the peroxisome proliferator activator receptor. It's key in storing, the gene is key in storing energy during food shortage and deprivation. So it's selected, it's very high frequency of the variant. Why? What environmental factors, let's say tens of thousands or hundreds of thousands of years, led to its increased selection? We don't know that. But the selected genetic variant leads to increased fat storage. That means if individuals does become changing lifestyle, then there is a higher chance that the variant leads to chronic diseases like obesity and other cardiovascular diseases. The environmental factor in the past that lead to selection, we don't know, but we speculate that based on the hypoxia-induced highland adaptation, it could be that partly. That's the Damota Tarara, Mount Damota. It's about 10,000 feet. It's not that high compared to the semi mountains, but compared to most places in the West, it's considered a highland. Diet is another factor. And the staple in the area, it's considered to be a threat against hunger. It's drought resistant. So perhaps it's an adaptive mechanism to store fat in case there is some shortage in the future, and that might have led to increased frequency of the variant. But future studies are required to understand this, and this has relevance for public health as well. So my last example in this piece will be podoconiosis. Colleagues in Bahir Dar University, Professor Wendemageng works on podoconiosis as well. He is my collaborator in the past as well. Podoconiosis leads to lower leg elephantiasis in genetically predisposed people who are barefoot in highland areas with irritant soil particles. And more than 4 million cases have been estimated in Ethiopia. It's an excellent model of gene environment interaction, where genetically predisposed people exposed to irritant red clay soil particles develop the disease. But earlier studies did not have any genetic information. So this was the focus of my PhD project. Now it's more than 10 years. So we designed a population-based genome-wide association study, recruiting cases of podoconiosis and controls in the Walaita zone. And GWAS, a genome-wide association study, is an approach where we scan hundreds of thousands or millions of genetic markers across the genome of many people to find association with a particular disease. It's like any case control study, except that you test it like millions of times, but it's a simple chi-square test. So with this approach, we successfully identified a variant in a locus on chromosome 6 known as the HLA in the cluster region that increases susceptibility to the disease. So at that time, published in 2012 in the New England, this was the first genome-wide association study of an uncommunicable disease in Africa, and it was validated by a family-based approach, HLA typing, and more recently, another PhD candidate, Tedros Tarifo, also validated the finding using research participants in the Amhara region and in Oromia regions. So With this strong evidence, polio was considered to be a T-cell-mediated immune response disease. So what's the relevance of this discovery? So our work, not just these GWAS, but kind of series of work we did, kind of characterizing the clinical grading of the disease, its social impact, and also the genetic discovery, helped increase the profile of the disease globally. So this body of research facilitated the recognition by the WHO of podoconiosis as one of the neglected tropical diseases. And using family health history as a tool, resources for podo prevention were targeted. Children with family history of the disease. This was a direct translation of genomics to public health, where the goal is to create a generation free of podo, where resources are provided to children who have an affected family member in the past, where using family health history. So lastly, I would like to add pharmacogenomic benefits of genomics. Pharmacogenomics involves assessing an individual's genome and targeting specific drug treatments to that individual. This is important because, as we know, drug response rates are variable. So it can be as low as 20%. Oh, 20%, only 20% of people prescribed a drug respond. And adverse drug reactions are also high. This is partly because there is difference between human populations for variants associated with function of genes involved in function of drugs like absorption and metabolism, as well as excretion. So one point I want to leave with you is that the label African or Black can be disastrous sometimes, and it can mislead clinical decisions because it ignores the genetic variation landscape within the continent. So we have seen that there is huge variation in the continent. So as an example, which is relevant, is our work documenting the distribution of pharmacogenomically important variants in Ethiopia. We did that in 2014, and we compared that to other African ancestry populations in the HAPMA. We found major differences between Ethiopian and other African populations in the data set in terms of distribution of these variants that impact response to drugs, as well as adverse effects of medications, such as for treating dyslipidemia, thrombosis, and myocardial infarction. So another important example is, again, from Ethiopia, is this study in 1996 by Professor Eleni Aklilu, now at the Karolinska Institute, showing that there are high proportions of Ethiopians who carry 2 copies of the cytochrome gene variant that metabolizes codeine. These individuals are known as ultra metabolizers because they are able to metabolize codeine, which is a commonly prescribed pain relief drug, to metabolize codeine to morphine very quickly. So some of these individuals would experience serious adverse reaction, especially in children, including respiratory depression and death. So the variant has one of the highest prevalence in Ethiopia, 30%. So in other Africans, including in African Americans, it ranges between 3 and 7%. We also validated this in our sample, that prevalence is about 29%, close to 30% in Ethiopia. In Europeans, it's 4%. Asians, up to 2%. In the Greek, a little bit higher. So in the Arabs, it gets a little bit higher. So what does this mean? So the FDA discouraged the use of the drug to children under 12 who are ultra metabolizers because of its consequence to respiratory depression as well as death. And I think this has even stronger relevance to Ethiopia. And I think the Ethiopian Drug Administration followed suit in the past, if I'm not wrong. So this is a great example of the importance of population-specific studies to make public policy decisions that have huge public health impact on a population, rather than borrowing recommendations from other places. For example, if we just use the African population data, then would assume that there are fewer ultra metabolizers in Ethiopia, but the data from Ethiopia shows that the highest prevalence of ultra metabolizers are found in the country. So what will happen if Africa remains fully represented in genomics? I'll end my talk with this. So this is a picture, which I call it green picture. A green picture. Over the past 15 years, hundreds of genome-wide association studies have been conducted. Several millions of individuals have participated in these studies. And when you look at the breakdown of the study participants by ancestry, more than 90%, more than 95%, 97% of the study participants are of European origin. Lack of representation of diverse populations will result in equitable access to precision medicine, and it narrows the broader societal impact on public health. So genomic technology is driving some of the most groundbreaking research, including precision medicine initiatives. But you still see it? Okay. But its full potential is not realized. So these are the largest population precision medicine initiatives. So you see Asia, Australia, Canada, the US, Europe.
It's unshared. Yes, Basil.
Oh, I'm sorry.
Just Andre, and the second line.
Okay. Sorry about that. Do you see it?
Yes. Thank you.
Okay. So we don't see any African country here where there are large population precision medicine initiatives. So if Africa is not represented, it's a missed opportunity to generate knowledge. So lots of information diversity means there is more information waiting to be mined, but because of the diversity within the continent is tremendous, it has an investment and economic benefits. So and also innovation implication, we lose that. Second, medical benefits from genomics may remain limited only to populations who are represented in research. So we have got a lot of lessons from genome-wide association studies who were conducted predominantly in European ancestry populations over the past 15 years. Now, when we start to see we combine them together and generate a polygenic score to see the cumulative genetic effect. And we try to predict that whether the polygenic scores are predictive of disease or other outcomes. It has been found that variants discovered in European populations are less predictive in African populations, which means when this tool is implemented in clinical practice, in public health policy, then African ancestry populations wouldn't benefit. So that would widen health disparities as well. Wrong clinical decision. This has implications not just for Africans, but for the whole world, because we have gotten some lesson from variant misclassifications for some diseases in the past, because this is an algorithm where we classify different exonic or pathogenic variants based on their allele frequency in human populations. So the assumption is the less frequent they are in human population, and they kind of fulfill some other criteria, we consider that as pathogenic. So in the past, there are examples where variants was considered to be pathogenic, and people with those variants were given drugs, as well as they were told to interrupt taking some drugs because they have that pathogenic variant. But that decision was based on studies that did not involve African populations. When African populations were involved, it was found that no, it's a normal human variation, not pathogenic. So that leads to wrong clinical decisions, and that decision could affect any individual, irrespective of ancestry. So what needs to be done? I'll just repeat what the WHO Science Council reported recently. Collaboration is important. There are some examples of successful genomic initiatives in Africa. The X-ray Africa project is one. Ethiopia institutes from Ethiopia have participated in tuberculosis project, the kidney disease, the neurogap psychosis program, where Ethiopia, Kenya, Uganda, and South Africa are participating are examples. And South Africa is a good example for implementing genomic technologies during the COVID-19. That's important. Secondly, capacity building. This is the recent publication based on the recommendation of the WHO Science Council on accelerating access to genomics for global health. I encourage you to read the report. The council recommends that, I'll read the quote, current uses and future applications of genomic technologies are critical for improving the health and livelihood of people in all parts of the world, regardless of economic status. And they put forward some recommendations, including promoting genomics through advocacy, implementation of technologies, and attention to ethical issues. The other important piece is country-led investment, commitment to investing in genomics as well as research and development. I think this is very important in addition to collaboration, because there is a commitment by African governments to spend at least 1% of their GDP on research and development to increase innovation. So this is critical to make sustainable impact in terms of genomics in public health, because it's not a wasted investment. I just wanted to give one example from the US. A current assessment shows that in the US, for every dollar in the US federal government investment on genomics, there is more than fourfold return. For example, 3.3 billion USD has been invested on genomics in 2019. The return was more than 15 billion USD. The point I want to make here is that genomics is no longer a luxury science, as some may think, but a daily health reality. It's also an investment opportunity for countries. So country-led investment on research and development is critical. So lastly, for sustained usefulness, it's also important to integrate genomics in public health and clinical curricula. As Professor Gaspari mentioned, Bahir Dar University recently started the public health genomics program, which is great. It's also one of the recommendations of the WHO Science Council, developing well-designed and integrated public health and professional education efforts nationwide. So I applaud the School of Public Health taking this initiative, and I hope it will help other universities also do the same in the future. So with that, I would like to acknowledge several people. What I presented is the work of several people. Some are here, and some maybe not here, but I would like to take the opportunity to thank my mentored fellows, trainees in my group, NICHD and NHRI, as well as my former mentors, current mentoring committee members, as well as collaborators. Thank you for your attention, and I'll be happy to take questions.
Wow, that was wonderful, Fasil. Really very, very impressive, from the very basic to the application and future and what we can do as an Ethiopian. So really appreciate that. So I'm sure there will be a lot of discussion. So whoever wants to start, please, you can unmute yourself or raise your hand. I like the idea of variation means information. So we Ethiopians, there's variation in all sorts of measurements. So that variation is important, but it's also depends how we use that variation or information. So thank you for that. Anyone who wants to start, please go ahead. We have a few minutes. Or you can put your question in the chat box if you don't want to talk. ሁሉም ግልጽ ነው። በአማርኛም በእንግሊዘኛም ይቻላል። ያ ሁላችንም እንሰማለን እዚህ ያለ ነው።
I went over the time. ይቅርታ ጠይቃለሁ ለዛ።
በጣም ጥሩ ነበረ። ለዛ ነው I think ሁሉም ሰው ብዙ ጊዜ ይወጣል ይገባል፣ ግን ዛሬ was stick together, so it was really good. ጥያቄ ካላችሁ ወይንም አለበለዚያ ያው እንግዲህ
if you have any questions, then feel free to send me an email.
Hello.
Maybe I'll put that in the chat.
Dr. Alam Sahay?
No, it's me, Barrow Gize. I mean, it's short Bell.
Yeah, go ahead.
Thank you very much, Fasil. It was really very insightful. I mean, I had a very little knowledge what has been done so far in a kind of African genomics, more specifically also Ethiopia, and then that's really very great initiative and work what you have been doing with your colleagues as well. So yeah, you were like focusing on the variation basically in terms of the population in Ethiopia as well. So besides all these developments, I mean, I do have some background challenge in terms of working with these admixed individuals. So how we can also integrate like the methodological development, whatever we are doing, GWAS analysis or other genetic analysis in this highly varied population. I mean, the tools what we have in bioinformatics, can we also be work on that?
Yeah, that's a great question. So thinking about the diversity of the Ethiopian population, the genetic variation of the Ethiopian population. So as you say, I'm not speaking in terms of ethnic differences. I'm speaking in terms of genetic variation, even among individuals belonging to the same self-identified ethnic groups. So as you said, the variation could be challenging in some instances in terms of methodological approach, because individuals could be multi-way admixed in your study population. So there are methods for that, for three-way and multi-way admixture, where you could study the admixture proportion and local ancestry association. But methods development in terms of, for example, 4 or 5-way admixed populations is still limited. There are some methods developed by colleagues, including Professor Tesfaye on admixture. So you can use that. But in general, in the grand scheme of things, if, for example, in our work, in our GWAS, in using some populations from the Walaita, genetic admixture was not an issue in the GWAS.
Okay.
Admixture becomes an issue when you have people from more divergent groups. It doesn't mean there isn't refined population structure. There is, but that is not strong enough to impact the association that you do. You can control that using, for example, principal component adjustment, as well as removing extreme outliers from your data sets, which basically would be few individuals. So I would advise that. So we had that. We also had data from the Oromia and Amhara regions for podoconiosis, and as much as we say there are refined fine level population variation, the variation that you see within each population group may be not much less than the variation you see between populations. So with good principal component adjustment and accounting for local ancestry, then you could control that.
Okay, thank you.
Very good. Thank you very much. Is there any? I saw Dr. Alam Sahay. Are you still there?
Hello.
Okay, yeah, go ahead. Kati.
እጅግ በጣም ነው የማመሰግነው። ፕሬዘንቴሽኑ በጣም የሚስብ የማይሰለች ሰዓቱ ሲደርስ ሳይታወቀን እንደደረሰ ነው የብዙዎቻችን አደርገው ስለሆነስ ፕሮፍ ተስፋዬና ዶክተር ፋሲልን እጅግ በጣም እናመሰግናለን እንደው ከዚህ ፕሬዘንቴሽን የተረዳሁት ይሄ ጀኖሚክ ጉዳይ በጣም አስገራሚ ያልተሰራበት ለወደፊቱ ደግሞ ትልቅ ኮንትሪቢዩሽን ያለው በተለይ በጤናው በፐብሊክ ሄልዝ ትልቅ ሮል ያለው እንደሆነ ነው አንደርስታንድ ያደረኩት እጅግ በጣም ደስ የሚል ነው እንግዲህ በየቢሮአችን በየቤታችን ሆነን እንደዚህ አይነት ትምህርት ማግኘት መቻል እጅግ በጣም መታደል ነውና ታንኪዩ ማለት ፈልጋለሁ ሁለታችሁንም ከዚህ በተጨማሪ እንደው መጠየቅ የምፈልገው ጠቅለል ያለ ጥያቄ ይሄንን የጀኖሚክን ፕሮግራም ፕሮፍም አንተ እንደ ካሪኩለሙን እንደቀረጽክልንና እንደተሰራው ፕሮግራሙን ለመጀመር አሁን ኦልሞስት ጨርሰናል ማለት ይቻላል ዝግጅቱንና ምናልባት ዶክተር ፋሲልም እንደው የምታግዘን ወይ ዳይሬክሽን የምትሰጠን ነገር ይሄን ፕሮግራም ኢምፕሊመንት ማድረግ ስንጀምር ያው የኢትዮጵያ ነባራዊ ሁኔታው ይታወቃል ኢኮኖሚው ምኑም ምናልባት ስታንዳርዱን በተጠበቀ አይነት ላብራቶሪ ወይንም በተደራጀ አይነት ሴትአፕ አይደለም እንጀምርና እንደው ምን አይነት ቻሌንጅ ይገጥመን ይሆን ይሄን ፕሮግራም አሁን እንግዲህ ከመስከረም ጥቅምት ጀምሮ እናስኬዳለን ብለን ነው የምናስብና አሁን ተማሪዎቻችን ሪሰርች እንዲሰሩ ይፈልጋል መሰረታዊ ነገር ስለሆነ ይሄን ስናደርግ ግን እንዳልኩት በተጠናከረ ስታንዳርዱን በተጠበቀ ላብራቶሪ አይደለምና እንደው ፐብሊክ ሄልዝ ኮሚኒቲው ላይ ምናልባት ዊዛውት ላብራቶሪ የሚሰሩ ብዙ ኤሪያዎች ይኖሩ ይሆን ተማሪዎችን ሊያግዙ የሚችሉ እንደዛ አይነት ቶፒኮች ይኖሩ ይሆን ነው እንደው ምን አይነት ቻሌንጅ ይኖራል እንደው ጠቅለል ባለ መልኩ ዶክተር ፋሲል አንተ በዚህ ኤሪያ ብዙ ስለሰራህ ምን አይነት ቻሌንጅ ይገጥመን ይሆን እንዴት ብንዘጋጅ የተሻለ ይሆናል በተለይ አንድ ባችና አንዲስ ከመናስጨርስና እስከምንጠናከር የሆኑ ቻሌንጆች ይኖራሉ ብዬ አስባለሁና እንደው ከሱ ጋር የሆነ ነገር ብትለን ደስ ይለኛል ሌላው በጣም ያስገረመኝ ፋይንዲንጎችን ስታቀርብ በአፍሪካ ነው በጣም ትልቅ ቫሪዬሽን እንዳለ እየነገርከን የነበረ ከሌሎቹ ዓለማት ሲነጻጸር እንደው የነበረኝ ሐሳቤ አፍሪካ የሰው ልጅ መገኛ ነው እንደዚህ ነው ምናምን እና ተመሳሳይ ሪላቲቭሊ ተመሳሳይ ጄኔቲክ ቫሪዬሽን ወይም ምስል የሆኑ ነበር ኤክስፔክት ያደረኩት ግን ከሌሎቹ ሀገራት ጋር ሲነጻጸር ይሄኛው በጣም ብዙ ሆኗል የሰጣቸው ትንታኔዎች በጣም ደስ ብለውኛልና አንዳንዴ በግምት ይዘናቸው የምንሄዳቸው ነገሮች ሁሉ ትክክል እንዳልሆኑ እንደዚህ በምርምሮች ዲስኳሊፋይድ እየሆኑ መሰረት እየያዙ የሚሄዱበት የትምህርት ዘርፍ እንደሆነ ነው አንደርስታንድ ያደረኩትና እንደው በብዙ መልክ እጅግ በጣም ደስ ብሎኛል ኢቭን ስለ ዲስኮርዳንት ስታወራ ራሱ ኤችአይቪ መጣብኝ ኤችአይቪ ዲስኮርዳንት እዚህ ጋር የተገናኘ ይሆን እንዴ ሚል ሁሉ ነገር እያሰብኩ ነበርና በጣም አስገራሚ ሳይንስ ያልተነካ ብዙ ሊያግዝ የሚችል እንደሆነ እንደው አንደርስታንድ ያደረኩኝ ታንኪዩ ስለ ቻሌንጆች እንደሆነ ነገር ብትለን ደስ ይለኛል
አመሰግናለሁ ዶክተር አለምፀሐይ ስለጋበዛችሁኝ በጣም ነው አመሰግነው ፕሮፌሰር ተስፋዬንም ጭምር ያ ፐብሊክ ሄልት ጂኖሚክስ ምን እንግዲህ ካሪኩለም አዲስ ፕሮግራም ስለሆነ ምን ቻሌንጅ ያጋጥማል የሚለው ጥቂት ነገር ማለት ጀመረ ቻሌንጁ ያው ብዙ ነው ኢትዮጵያ ውስጥ ብቻ ሳይሆን ኢቨን ሪሶርስድ ቦታዎች ላይ ለመጀመሪያ ያህል ለምሳሌ ቻሌንጆቹን ለመወጣት ምን ማድረግ ይቻላል በተለይ ሪሰርች ኢንቫይሮንመንት ላይ አንደኛው ኮላቦሬት ማድረግ ነው አዲስ የላብ ዴታ ጀነሬት አድርጎ ተማሪዎችን ቶሎ ያን ሁሉ ፕሮሰስ እንዲያልፉ ለማድረግ ሊከብድ ይችላል የዌት ላብ ሴቲንግ ላይ ነው ይችላል ስለዚህ አንድ ማድረግ የሚቻለው ኤግዚስቲንግ ዴታዎች ይኖራሉ ኢትዮጵያ ውስጥ ኮላቦሬት ማድረግ እነኚህን ኢንስቲትዩቶች አይደንቲፋይ ማድረግ within the country ለምሳሌ የአዲስ አበባ ዩኒቨርሲቲ የኢትዮጵያ ባዮቴክኖሎጂ ኢንስቲትዩት I worked with AfRIE, Arman Hansen Research Institute. There are some data sets as well. እነኚህን ዴታዎች ለመጠቀም መሞከር ከዛ በተጨማሪ publicly available የሆኑ some of the presentation እኔ ያቀረብኩት publicly available የሆኑ ዴታ ሴቶች ናቸው እነሱን አክሰስ ለማድረግ መሞከርና እነኛ ላይ ሪሰርች ፕሮጀክት ዲዛይን እንዲያደርጉ ተማሪዎችን ማበረታታት አንዱ ነገር ሊሆን ይችላል ብዬ አስባለሁ። ሌላው ብዙ ጊዜ ፐብሊክ ሄልት እኔም ፐብሊክ ሄልት የተማርኩት ፈርስት ኤንድ ማስተርስ ዲግሪ ተማሪዎች ወይ ሁላችንም what's the immediate public health impact የሚል ጥያቄ ሁሌ እንጠይቃለንና ያንን አድሬስ ለማድረግ የሚያስችሉ ለምሳሌ LC issues ethical legal social implications of genomics ምንድነው ፖሊሲ ላይ ያለው ኢምፓክት ምንድነው እንደዚህ አይነት ሪሰርቾችን መስራት ይቻላል በተለያየ መንገድ primary data collection including qualitative studies we did that ለምሳሌ when I was a PhD student in the Walaita LC issues ነበሩበት የሰራናቸው ስራዎች ውስጥ ያንን መስራት ይችላሉ especially as a first program yeah we can come up lots of research questions ኢትዮጵያ ውስጥ scale up ለማድረግ ፕሮግራሙን ያሉትን ቻሌንጆች አይደንቲፋይ ማድረግ በራሱ it's a big research question እኛኛ ማድረግ ይችላል ሌላ public health genomics even genomics አዲስ ስለሆነ ምናልባት አድቮኬሲ ላይ ተጨማሪ ስራ መስራት ሊያስፈልግ ይችላል ብዬ አስባለሁ within the Ethiopian higher education system Ministry of Health ባይል ለማግኘት ስራዎችን መስራትና እነኝን ሰፖርት ሊያደርጉ የሚችሉ ግሎባል ስራዎች ለምሳሌ WHO on council report it supports what you are doing especially after the COVID pandemic strongly even irrespective of resource genomics implement መደረግ አለበት potential አለው this is the way forward below outline ያደረጓቸው ነገሮች አሉ ያንን ተጠቅሞ reach out ለማድረግ መሞከር ይቻላል ብዬ አስባለሁ እንግዲህ እኛም በሚቻለው መጠን ለማገዝ እንሞክራለን
በጣም ጥሩ ይሄ ፕሮግራም ሲዘጋጅ ፋሲል ዋናው ታሳቢ የተደረገው እሱ ነው I think ከለሱ የፐብሊክ ሄልዝም ግራጁዌት MPH አለው እንደገና ጂኖሚክስ ማውን በሙሉ በስፋት እየሰራ ነው ያለው እና I think ትልቁን ሮል ሊወስድ የሚችል አንዱ ፖቴንሻል ሪሶርስ ፐርሰን ነው እና እሱም ታሳቢ የተደረገ ነው እናመሰግናለን ሌላ ላስት ሚኒት ጥያቄ ወይንም ዲስከሽን ካለ I think we are ሰዓት እየሄደ ነው ጥያቄ ያለው ሰው አለ ወይንም ኮሜንት ለዲስከሽን የሚሆን
maybe comment አይደለም ፕሮፌሰር ተስፋዬ
yeah go ahead
okay maybe as I am working as a project development and management office ነው የምሰራው ባህር ዳር ዩኒቨርሲቲ እና maybe እግረ መንገዴንም ዶክተር ፋሲልን ያመሰግን that's new we do have energetic staff ሳለ ፕሮፍ ተስፋዬ እንኳን የተወሰነ ነገሮች አሉ ግን we lack skills and knowledge related with genomics አሁን እንዳለችው ዶክተር አለም ሳይም ማለት ነው so if you are የተወሰኑ ነገሮችን ዳይሬክሽኖችን help ካረከን ካሳየን በጣም የሚጽፉ ስታፎች አሉ ባዮቴክኖሎጂም አለ እዚህ ከኛ እንደ አንድ ሪሰርች ሴንተር አለ ኢቨን ምናልባት ፕሮፍ ሼር አድርጎ ሊሆን ይችላል ጥሩ ላቦራቶሪ ቢልዲንግ ተገንብቷል ለኢስት አፍሪካ የሚሆን ማለት ነው በጣም ከኢትዮጵያ ሁሉ ኦልሞስት ማለት ይቻላል ዩኒክ የሆነ ነገሮች አሉና ተስፋ ነገሮች አሉ እኛ ጋር ይሄንን ለመስራት ማለት ነው ስለዚህ we can write a grant for NIH እንደዚህ አይነት ነገሮችን ሪኩዌስት እናደርጋለን ስታፎችን ማስተባብረን አንተም ካለ ዳይሬክሽኖች እዚህ ተዘጋጁ እዚህ ላይ ብትለን አስተባብረን የተወሰኑ ነገሮችን እንሞክራለን ለማለት ነው ስለተዋወቅነ ወይም ደግሞ ስላቀረብልኝ በድጋሚ አመሰግናለሁ ይሄው ነው አለ አስተያየት
አመሰግናለሁ briefly ሼር ማድረጋቸው ነገሮች ናቸው ባህር ዳር የተሰራ ስላለው የባዮቴክኖሎጂ ኢንስቲትዩት ሰምቻለሁ I'm sure ሆፕፉሊ የሆነ ኦሪጂናል ላብ ወደፊት ይኖራል ብዬ አስባለሁ። ያ እንግዲህ ፕሮፌሰር ተስፋዬን ኮኦርዲኔት ስለሚያደርግ ወደፊት እንግዲህ አንዳንድ ሚቲንጎች አሬንጅ አድርገን አንዳንድ እንደዚህ አይነት ዲቴይሎችን ዲስከስ የምናደርግባቸውን አቅጣጫዎች ማንሳት እንችላለን ብዬ አስባለሁ።
በጣም ጥሩ። አይ ቲንክ ጥያቄው እንትን ሪኮርዲንግ እና ስላይዱ ሁሌ እንደምናደርገው ሸር እናደርጋለን። በዚሁ እንጨርሳለን የዛሬውን። እንኳንም ተገናኘን። የኔክስት መንስ ፕሮፌሰር ዮናስ ገዳ ስለ ኬዝ ኮንትሮል ለማን ኮርስ ስተዲ እና የመሳሰሉ ነገሮችን ያቀርብልናል። እንግዲህ እንገናኝ ለበዛን ቀን። መልካም ዓመት በዓል እስከዛ።
መልካም በዓል ለሁላችን።
እሺ ቻው።
መልካም ቻው።
Thank you.